Okay, I'm Chris Mundt. I'm a serial pathologist with Oregon State University, and I'm stationed down in Kalous on the main campus, but I try to cover serial diseases as best I can around the entire state. Jay?
Thanks, Chris. And I'm Jay Kalous. I'm the winter wheat breeder with LimaGrain cereal seeds based in Walla Walla.
I'm going to be talking a little bit about a disease called Cephalosporium Stripe, which is caused by a fungus, and it gets its name by this very wide stripe that you can see on this leaf here. And under severe conditions like we have in the field right now, you can see it's actually killing off this entire tiller. This is a trial here where we're dealing with Cephalosporium Stripe of winter wheat. In this case, LimaGrain was really interested in trying to find some DNA markers that are associated with resistance to Cephalosporium Stripe. And we know that some of the resistance genes associated with Cephalosporium Stripe also might have some impact on Fusarium crown rot. So we're hoping to get some information potentially on both diseases here. For both of these diseases, there isn't any one single gene that will give you a high level of disease. Rather, you have to accumulate a few or so genes together to try to get the kind of resistance that you want to see out in your field. So what we have out here is a trial where we have 400 different genotypes of wheat from the LimaGrain program, and they're replicated 4 times. We're not taking yield on these trials, we're just taking disease observations, so we can go for a little bit of a smaller plot. We plant early to try to make sure that we get good expression of Cephalosporium Stripe. So this was planted, I believe it was the 9th or the 10th of September. And then end of October, beginning of November, what we do is we artificially infest this field. Put about, I believe it was about 170 pounds of inoculum total out in this field, which is almost exactly an acre out here. Okay, so this is working out to be a fairly severe year for Cephalosporium Stripe in our inoculated trial here. I think it's a combination of the winter injury that we had along with the drought stress that everybody's experiencing right now. And normally this time of year I'm just starting to see some of the symptoms, but now already, and here we are mid-May, and we're starting to see some fairly severe symptoms. So all of these tillers that you see here that are short and yellow, those have all been nailed by Cephalosporium Stripe. And the only ones that are somewhat normal are these taller ones here. So it looks like we could have as many as 80% of the tillers actually killed off by Cephalosporium Stripe in this susceptible genotype here. What we'll do later on is we're going to go through and give estimates on each of these plots in terms of the percent of heads that have white heads. And we've found over the years that gives us the best indication of how severe Cephalosporium Stripe disease is. Then we turn those data over to LimaGrain, and Jay's going to talk about what they're going to do with it. But basically we have a collaboration whereby I do the phenotyping out here in the field, give them the data, their marker lab goes and tries to associate DNA markers with resistance genes, and then they share the closest associated publicly available marker and make that available to us. So it's very much a sharing kind of relationship.
So yeah, the hardest part with most diseases, I would say, is the phenotyping, the actual field note taken. It's very difficult to get a natural infection that's uniform across the trial. It's very difficult to separate out if it's more than one disease. So confounding effects, field effects. So being able to have Chris help us with this in a trial that there's a lot of uniformity for a field trial in terms of disease makes the data that much better. So what we'll do is all of these individuals that are out here, they'll be genotyped, and so we'll do across the entire genome, we'll put different tags of all the DNA throughout, and then we'll take the phenotype data and we can start to associate a marker and a region in that genome with resistance to Cephalosporium. And we can be confident that it is Cephalosporium tolerance because of the quality of the phenotype data. It's a complicated process. Essentially, it's kind of like The interstate highway system is how I think about it. So you have interstate roads all over the US, and they're divided up with mile markers. Those mile markers don't exactly tell you much of anything other than where the next mile marker is. So it's kind of a position where you are relative to another mile marker. And that's what I think of when I think of DNA markers. They're just telling you where you are relative to another marker within the genome, within the wheat genome. But when you start to couple that information with the phenotype data, that's when you start to say, okay, well, at this mile marker, there's a gas station. And at this mile marker, we've got a restaurant, and then there's also a hotel there. And you can think of that as like the genes of interest that you're after. So next to this marker, there is a cephalosporium resistance marker. Because we know that ceph is quantitative, we want to add some diversity so we can start to pick up, we're going to need maybe 6 genes or 7 genes, and all of those combined is going to give us our resistance to ceph. And you can't pick that out when it's just a resistant versus susceptible line. By adding the diversity, we're hopefully going to be able to uncover a haplotype, so a collection of alleles that give the desired phenotype.
Jay, can you tell folks a little bit about the diversity of germplasm out here, how it was chosen, etc.?
Sure. The material here is essentially, it's all breeding material. It's material throughout our pipeline. So from Y1, so a first year yield line, all the way up to our most advanced material. We selected based on a lot of pedigree information. So we do know of some varieties that tend to have a little bit better ceph resistance, and we know of some varieties that tend to have a little weaker. And so we tried to get a collection of both types roughly in even amounts to aid in the likelihood of making those marker trait associations that can then transfer to the greater population of material. So the nice thing is that we did have a very active collaboration early on with Oregon State, and there was a lot of sharing of genetic material. And so what we find in this material should directly translate into the Oregon State program, and I would say probably even into any other public program in the region.
If we could have a situation, for example, maybe, Jay had a bunch of lines that just had really fabulous yield, but he wasn't sure which ones of them had really good disease resistance. Well, that's information that I can help him out with, and then hopefully we wind up with a variety that has good yield, good disease resistance that we can get out to the grower.
For me, whenever we do approach a collaboration, in my mind, the end goal always is the same, and that is to make a farmer more money. That's either with reduced inputs or on higher yield on the other end. So for us, this collaboration is just a great opportunity. There's a wealth of knowledge and resources at Oregon State, and just to be able to work with Chris and Christina is fantastic opportunity. We feel it's just a great advantage just to tap into those resources in terms of pathology at Oregon State.